So did you all enjoy your weekend? It's always kind of disconcerting to go to class for a week and then have a 3-day weekend.
Let's do it again.
How many of you have read your lab? Okay.
Not that far.
Read it tonight. Read it tonight. What we're going to do is there's a fair amount of kind of, especially when you make media, waiting for stuff. And so what I'm going to do is I'm going to have this table start out on the microscopy section, and this table start out on making the media and those exercises. And once you finish making your media, you can do the microscopy, and then you guys can do vice versa. So I'll have a couple instructions when you come in tomorrow, but otherwise, go ahead and get started. Okay. And Kelly, you're working with this group right here, correct? Okay, so we're doing again 2, 2, 2, 2, 2, or sorry, 3, 2, 2, 2, 2. Okay? Questions? Okay, we're making our run through this kind of composite bacterial cell, highlighting some of the structures that the bacteria may have. Again, please remember that when we go through this list of all this stuff, endospores, capsules, etc., not all bacteria will have all these structures. And as a matter of fact, what structures they have and your ability to detect those structures will be really important in being able to identify your unknown. And so these are species-specific characteristics. We've kind of started out from the outside looking at the external surfaces, some of the appendages. We were talking about the glycocalyx and ended up talking about the capsule a little bit. Remember that there are 2 major roles that the glycocalyx plays, and one is protection, and that's particularly true of the capsules. Protection from what? Desiccation and phagocytosis, particularly with pathogens, okay? But they also play a role in attachment, and we talked about the fact that both the glycocalyx and the fimbriae can play a role in organisms sticking to each other and to host cell surfaces. And that oftentimes is a real important step in pathogenesis and organisms causing disease, because unless you can gain a foothold on a tissue, the pathogen really can't invade that tissue. So attachment is really an important part of the strategy that a pathogen has to be able to invade tissues and a host. Let's talk a little bit about biofilms. This is one of those interesting topics that even probably 5 years ago, I really wouldn't have been mentioning at all in this particular class. This idea of a biofilm is really kind of a relatively new concept. Really what a biofilm is, it's a community of microorganisms living together, interacting with one another, and there really has built up a whole new discipline of microbiology called community microbiology, which deals with biofilms and their formation and their significance. We used to think of microorganisms as living fairly solitary existences. Now, true, microorganisms will be found in mixtures with other organisms. I said the other day that if I did a plate of some skin rubbings, I would get some staph bacteria, but I would get other species as well. But we used to think of those living kind of independently from one another. So the staph did their thing, and the corynebacterium did their thing. But now we're finding out that these organisms tend to attach to not only surfaces like the skin, but one another. And once they start forming this complex community, they start interacting with one another in fairly interesting ways. In nature, it's estimated that probably 99% of bacteria live in this kind of biofilm community. This slide gives you a very simplistic kind of introduction to how a biofilm might form. So here we've got up at the top some organic surface, some cell surface coating, for example. And here are our first colonial organisms, and they are able to adhere to that organic substrate by a glycocalyx or a fimbri or something. Those cells start adhering not only to surfaces but to one another, trapping cells and making kind of a conglomerate. And then many times these cells will start to secrete extracellular substances which further facilitate other cells becoming adherent to this mass. And pretty soon you get this complex community of different species all interacting together. Probably one of the biofilms that you're most familiar with is dental plaque. If you go to the dentist and get your teeth cleaned and they have to scrape off all of that calculus or tartar, that really is a biofilm. One of the reasons that sugar, particularly sucrose, promotes dental caries or dental cavities is that sugar is an important constituent in making that biofilm. It is incorporated into some of the external polysaccharides that make up the glycocalyx, and so you need that sugar to kind of get everything stuck together. And then what makes plaque so dangerous is that once this biofilm is formed, organisms start metabolizing the sugar as well, and when they ferment the sugar, they produce acid, and then the acid can decay the enamel. Biofilms are very commonly found in aquatic environments, and the biofilms can oftentimes clog sewage treatment pipes, for example. They can give materials that are exposed constantly to water this kind of slimy appearance, and it really is this very complex community of microorganisms. There is a lot of chemical signaling that takes place between the organisms of the biofilm. There is a phenomena that oftentimes operates in biofilms called quorum sensing. And quorum sensing refers to the ability of microorganisms to sense other microorganisms in their environment. And in quorum sensing, when a certain density of microorganisms accumulates, the cells are able to detect that, and it causes some real changes in their gene expression. For example, a lot of the earlier colonial cells that establish a biofilm are motile. And in their free-living state, these particular organisms express the gene for pilin to make pili, or the gene for flagellin to make flagella, because they're motile and they're moving. Okay? But once they settle down into this biofilm and start attaching to surfaces and one another, when they reach a certain density, they start self-signaling one another, going, hey, we're here. Okay? And those cell signals alter gene expression, and they turn off the genes then for flagellin and start making that particular protein, because they're stuck now. They're not going to be moving around anymore. And they start expressing different genes. Sometimes those genes code for substances called virulence factors. And virulence factors are things that help microorganisms to cause disease. So sometimes these genes aren't expressed until the bacteria is part of a biofilm. We think now that probably as many as 2/3 of infections involve microorganisms that are part of biofilms in the host organism that's infected. So studying and understand biofilms has some real important applications. Okay, this particular slide right here illustrates what I was just talking about right now. Here we've got some free-swimming cells that are settling on some kind of a substrate. They start to gather together and held together by this matrix. And then when the biofilm grows to a certain density or quorum, the cells start releasing these inducer molecules, like these green molecules right here, these quorum-dependent proteins. And that starts to change and alter gene expression. So really what happens is these cells now start communicating with one another chemically, and also they get kind of synced together. Okay? They might start producing new enzymes, for example, for digesting new food sources that are made available by the presence of the biofilm. For pathogenic bacteria, being part of this biofilm can actually be somewhat protective. So, for example, it can be difficult for antibiotics to penetrate this massive material that makes up the biofilm and get access to the microorganisms. Antibiotics are only effective if the microorganism takes them up, because it's got to be able to do something like inhibit cell wall synthesis for the penicillins or interfere with protein synthesis. So if the antibody can't gain access to the cells, it's not going to be effective. When you have a complex community of cells, as you do in a biofilm, then the chances are that one or more of those species of bacteria could be resistant to the antibiotic is increased. So even if the antibiotic can gain access, there's a greater chance that there might be resistant forms present. Also, oxygen has a difficult time penetrating into the mass of the biofilm. Now, you might think that wouldn't be good for the bacteria. But many bacteria have the ability to carry out a type of respiration called anaerobic respiration. These organisms are called facultative anaerobes. So they can carry out good old aerobic respiration when oxygen is present, but shift over to facultative if oxygen isn't present. So they can survive in low oxygen concentrations. We're going to see later on that some cells use oxygen, and particularly the toxic byproducts that are produced from the metabolism of oxygen, in killing mechanisms. Phagocytic cells have these oxygen-dependent killing mechanisms. Oxygen is metabolized and produces things like peroxides, and peroxides are toxic to the pathogen. Well, phagocytic cells, even if they can gain access to the cells in this biofilm, if they're deprived of oxygen, that will knock out these oxygen-dependent killing mechanisms and therefore eliminate one of the strategies your body uses to try to deal with pathogens that have infected your tissues. So there can be some real advantages to pathogens existing in these biofilm communities. So here's a couple of examples of biofilms that are not uncommon. It's a little bit hard to see right here, but there's a catheter back here, and these are both fungal cells and staphylococci that are adhering to this catheter, and therefore if the catheter isn't properly cleaned and is used again, then those cells can be transmitted into a new patient. Here is a biofilm developing of staphylococcus on heart valve tissue, causing then heart disease. Okay. One of the more complicated cellular structures is part of the next kind of layer of cell structures, the cell envelope. And the cell envelope is going to consist of usually a cell wall and then absolutely a cell membrane. Okay? You've got to have a membrane to be a cell. Most bacteria, virtually all of them, have a cell wall. There is an exception, and we'll talk about it a little bit later on. There is a type of bacteria called mycoplasms which do lack a cell wall. However, there is variation in structure of the cell wall, and we'll talk about that variation, but particularly right now, we're going to be interested in variation that results in two categories of bacteria, gram-positive cells and gram-negative cells. And this is one of the very first staining techniques you are going to learn is the gram stain, and the gram stain will be a very important tool throughout the course of the semester and critical in identifying your unknown. I can tell you right now, when we have our first lab exam, I normally don't make you memorize all of the steps in a particular staining procedure, with the exception of the gram stain. And I can tell you right now that during the lab exam, there will be a couple of stations set up right here, and there will be some tubes of organisms, and you will have to do a gram stain on them, and do the stain, and then look at it with a microscope, and report the gram reaction and the morphology of those cells. It's such a fundamental technique, you're going to do it over and over again. It will become second nature to you, and that's what you should just be able to look to without having to look at instructions. You may know what the purpose of the bacterial cell wall is?
Structure.
Elaborate. What do you mean structure?
Like rigidity.
Okay. So it does give the cell rigidity. It can and does protect the cell from lysis, particularly if the cell is in a hypotonic environment. Remember the terms hypertonic and hypotonic? What happens to a cell when it's placed in a hypertonic environment, a salty environment? It shrivels, okay? Water's pulled out of the cell by osmosis. What happens to a cell when it's placed in a hypotonic environment? It swells, okay? And it builds up internal pressure. Remember what that pressure is called? Turgor pressure, okay? And the cell becomes swollen or turgid. And if that cell wall weren't present, what would happen to the cell? It burst or it lyse, okay? So the bacterial cell wall has a real role that it plays in terms of preventing cell lysis from occurring in hypotonic environments. Do you know why penicillin works? It prevents cell walls from being resynthesized. Now, what does that imply? That implies that cell walls are broken down, right? Why would a cell wall need to be broken down? In order to allow stuff to come in, that would be true, but the cell wall, as it turns out, really isn't much of a barrier, particularly the gram-positive cell wall. Gram-negative, we got a little bit more of an argument there. How do cells divide? By what process? Remember what we called it? Cytoplasm splits in half? Binary fission? Okay. So what would happen each generation of division if we just had binary fission? They get smaller and smaller and smaller, right? Okay. So what has to accompany binary fission if we're going to maintain viable cells? Cells have got to enlarge. Okay? So that this cell basically grows and achieves the size of the original parent cell. Same for this. Same for these. Okay? Well, the way that bacterial cells enlarge is by selectively loosening their cell wall so that when water comes in, the cell swells and stretches. But then they resynthesize that cell wall before the cell gets so turgid that it bursts. So the cell's in this dynamic process of having to temporarily break down the cell wall or loosen it to allow stretching, okay, kind of like having elastic pants, okay? And then at some point in time, it's got to tighten it up again in order for the cell to survive and not burst. Okay? What penicillin does is penicillin gets into the cells and prevents the resynthesis of that cell wall. So it loosens, but then it can't reform again. So what do the cells do? They eventually lyse and burst. That's how penicillin works. Another thing that cell walls do is they help to determine the shape of the microorganism. And we'll get into that in just a little bit when we talk about cell wall structure. Does anybody know what the enzyme lysozyme does? Don't confuse it with the organelle lysosome, but there's an enzyme called lysozyme. It's found in your tears. It's found in saliva, two places where bacterial infection could not be uncommon if it weren't for the lysosome. Because your eyes are exposed to dust and dirt. You put all kinds of things in your mouth. One of the protective mechanisms you have is that your body secretes this enzyme lysosome. It breaks down the material that makes up bacterial cell walls. Okay. Well, if you take a cell and you digest it in lysozyme in an isotonic solution, if you take a bacillus and you take a cocci and you expose them both to lysozyme and digest away the cell wall, they both become spherical. Okay. So the shape of the bacillus is due to the constraints imposed by the structure of this cell wall. So different morphologies are due to different cell wall organizations. What are plant cell walls made of? They're made of cellulose. They're polymers of glucose that make up these cellulose microfibrils. And then there's some other interesting stuff in plant cell walls. There's some pectins and things like that. But it's mainly polymers of cellulose. Are bacterial cell walls made of cellulose? Nope. They're made up of a structure called peptidoglycan. So bacteria have peptidoglycan cell walls. We'll get to peptidoglycan in just a second. But first, let's tackle this idea of what is different in these cells that we call gram-positive cells and what is different in those cells that we call gram-negative cells. Now, operationally, it's pretty simple. We do something called a gram stain, and I'll go through the gram stain in a little bit. But at the end of the gram stain, gram-positive cells stain purple, and gram-negative cells stain pink. The question is why? Well, this is a reflection of some fundamental differences in the way that the bacterial cell wall is organized in the gram-positives and the gram-negatives. Now, they both have peptidoglycan. They share that in common. But one of the things that we'll see is that the amount of peptidoglycan is much different in gram-positives and gram-negatives. These have a big old thick layer of peptidoglycan. These have a very thin layer of peptidoglycan. But where the real difference comes into play is that these gram-negative bacteria have basically a second membrane called the LPS layer. Okay, so again, if you're a cell, you've got to have a plasma or a cell membrane. If you're gram-positive, stuck on top of that is a big old thick layer of peptidoglycan. If you're a gram-negative, stuck on top of the cell membrane is a little thin layer of peptidoglycan. And then a second membrane, the LPS layer. These cells have kind of a double membrane. So here's a micrograph of the cell over here on the right, and then a little kind of diagram showing how things are. put together. You can't see this well on the micrograph. So here's your cytoplasm, okay? And here's your cell membrane. And then we have this big old thick layer of peptidoglycan. We don't know what peptidoglycan is yet. We will get there in a second. In addition to peptidoglycan, which makes up about 90% of the mass of the cell wall, there are a couple of other important molecules associated with the gram-positive cell wall. There is teichoic acid and lipoteichoic acid. So this is a little bit easier to see over here if you look at this. Okay, so here's our gram-positive cell wall over here. Cell membrane, good old phospholipid bilayer, and then stuck on top of that is this big massive layer of peptidoglycan. And then inserted into the peptidoglycan are these teichoic acids. The lipoteichoic acids tend to span the layer of peptidoglycan pretty much entirely and actually insert themselves down into the cell's plasma membrane. These particular molecules both have a net negative charge, and that contributes to the overall net negative charge that most bacterial cells have. In a couple of weeks, I guess next week, you're going to do, 2 weeks, we're going to do some simple staining. And one of the things that we're going to do is we're going to expose bacterial cells to positively charged stains, like crystal violet and safranin. Those are two we use in the gram stain. And those stains, the positive charge of the stain will bind to the negative charge of the cell. And this is where a lot of the negative charge is coming from, is from these teichoic acid molecules. These particular molecules also can be very important antigens. What is an antigen? I've all heard the term before, I know. You might not know what it is. Antigen is short for antibody-generating molecule. These are substances that stimulate the formation of antibodies. Many times they're referred to as being foreign, but they don't have to be foreign. There are some instances where the antigens of your cells stimulate your immune system to form antibodies against your own cells. What do we call that? Autoimmunity. Okay, there's a lot of diseases we're finding out now that are the result of autoimmunity. Normally, though, what happens is your immune system is reacting to molecules it's not used to seeing, to these foreign antigens. And one of the major foreign antigens we find on gram-positive pathogens are these teichoic acids. So this is what antibodies are reacting to. They're binding to these particular antigens and then helping to eliminate those from your body to protect you from infection. There are some other proteins that are part of this gram-positive cell wall structure. And some of these proteins can be toxic to host cells. You probably all have heard of Streptococcus before. There are several different Streptococcus species that can produce diseases. And strep bacteria have a protein associated with their gram-positive cell wall structure, which is toxic to human cells and responsible for some of the disease symptoms associated with strep infections. Now, I don't know if you can see up here, but there's this little box right here that says periplasmic space, and it's got a little arrow that goes over here to the gram-negative cell wall structure, and one that goes over here to the gram-positive as well. This seems to be a real point of contention among authors of books and publications. Many of the books that I've read and seen say that the gram-positive cell wall structure doesn't have a periplasmic space. Others, like this slide, say it's got a little tiny periplasmic space, the space between the cell membrane and the peptidoglycan layer. I think more often than not, I've seen it indicating that they don't consider a gram-positive cell wall structure to have a periplasmic space. It's definitely a feature of the gram-negative cell wall structure. Okay, so here's our gram-negative cell wall. So again, here's our cytoplasm over here. Here's our cell membrane. Then we have this well-defined periplasmic space, peptidoglycan layer, and then in green we have this outer second membrane called the LPS layer. And that's really distinctive and very unique. In gram-negatives, the peptidoglycan only makes about 5 to 20% of the total cell wall mass. So much less extensive than it is in the gram-positive bacteria. So, over here on the right, here's your cell membrane, here's your thin layer of peptidoglycan, and then on top of that is this LPS layer. The LPS layer is a phospholipid bilayer like the cell membrane is. But associated with it are these lipopolysaccharide molecules. That's what LPS stands for. It's lipopolysaccharide. Some of them are confined primarily to the surface of the cell and kind of stick out like little tails from the top of the membrane. And others penetrate down into the membrane and actually anchor this LPS layer to the peptidoglycan. One of the lipopolysaccharides has a particular type of lipid associated with it called lipid A. And lipid A is a toxin. This particular type of toxin is called an endotoxin. The lipid A isn't released unless the bacterial cell dies and the LPS layer starts to break down, releasing these lipid A molecules. So bacteria that can cause this endotoxin poisoning don't have to be alive to make you sick. As a matter of fact, they're more likely to make you sick when they die and the lipid A is released from these particular cells. Lipid A is kind of a nasty substance. It won't kill you, but it'll make you sick enough that you're sitting there wishing, I wish I could die. Fever, nausea, vomiting, diarrhea. One of the gram-negative bacteria that is infamous for producing lipid A toxin is Salmonella. What is Salmonella toxin? Pardon me? What does Salmonella cause? You ever heard of Salmonella before? Food poisoning. Okay. So if you've suffered from Salmonella food poisoning, it is the result of the breakdown of these Salmonella cells, probably by your immune system, the release of the lipid A molecules, and then they get into your circulatory system and start affecting the intestinal cells, epithelial cells, etc., and causing those particular symptoms. The periplasmic space is kind of an interesting location. So it's this region really between the outer membrane and the LPS layer. And in this periplasmic space, there are a lot of digestive enzymes. So that food that is brought into the cell across the outer membrane is kind of held in this reservoir, and then the bacterial cell secretes digestive enzymes, and then it's broken down into a small enough set of molecules that it can then move across the cell membrane into the cytoplasm. I said that the cell wall isn't much of a barrier normally to getting things across the cell surface. Peptidoglycan itself is a pretty loose-knit structure, and even fairly large molecules can move across that layer. Since a gram-positive bacterium is primarily just a thick layer of peptidoglycan, there's really not much filtering that takes place by the cell wall. But the LPS layer is like a cell membrane in that it is pretty selective, and only certain things can get across it. It actually is leakier to most substances than the plasma membranes. And that's because embedded into this LPS layer are a whole bunch of transport proteins called porins. And there are 30 plus of those porin channels that have been identified so far. And they can bring a wide variety of substances, including some fairly large molecules, across that first barrier into the periplasmic space. And then if things need to be digested a little bit, they can be broken down by enzymes, and then those products can be brought into the cytoplasm of the cell. The first form of penicillin that was discovered, a naturally occurring form of penicillin. You may know what produces penicillin, where it comes from. It comes from a mold, okay, Penicillium notatum. And the naturally occurring form of penicillin is a molecule called penicillin G. And then drug companies have modified that penicillin to produce all kinds of penicillin derivatives. Penicillin G, the natural form of penicillin, really only works effectively against gram-positive bacteria. So it's great for treating staph infections because staph is gram-positive. Why doesn't penicillin G work on gram-negatives? Gram-positive is more penetrable, correct. The gram-negative LPS layer basically blocks penicillin from getting into the peptidoglycan layer and inhibiting its resynthesis. The derivatives of penicillin that have been produced, though, have been tweaked enough that they can cross that LPS layer. Those particular derivatives of penicillin, like amoxicillin, I bet everybody said amoxicillin at some point in time, right? Get rid of ear infections, probably amoxicillin. Works against both gram-positive and gram-negative. Penicillin is said to be a narrow-spectrum antibody. Amoxicillin is what type of antibody? Well, a broad-spectrum antibody, because it can work on both gram-positive and gram-negative. So this is a good slide that does a good comparison of gram-positive and gram-negative cell walls. And again here, this author tends to say a periplasmic space is present in both gram-positives and gram-negatives, that it's very narrow here. Again, some other authors say no, there's no periplasmic space. Okay, let's take a look at peptidoglycan for a second, because it's common in both gram-positive and gram-negative cell wall structures. Peptidoglycan is a polysaccharide polymer. Okay, so it's got that in common with cellulose. But one of the things that's different between this and cellulose is that the polysaccharide polymers are hooked together by amino acid bridges. The two sugars that make up the polysaccharide chain are abbreviated NAG and NAM. NAG stands for N-acetylglucosamine. NAM is N-acetylmuramic acid. And if you're interested, you can see that in these particular NAM sugars right here, you can see that there is an amino group, there's an acetyl group here. N-acetylglucosamine is not found just in bacteria. Actually, N-acetylglucosamine is also the primary constituent in chitin. So chitin is? We find chitin. Exoskeletons, okay, arthropods, insects. NAG, yeah, it's found in chitin. NAM, N-acetylmuramic acid, is unique to prokaryotes. Now, this is a little bit hard to see up here. But if you look at this middle box right up here, okay, so here's your peptidoglycan sheet of material. And if you start breaking it down and looking at it, there are alternating sugars in these chains of NAMs, the purple, and then NAG, the yellow. So peptidoglycan is made up of these chains of NAG, NAM, NAG, okay? But then each of these chains is cross-linked to another by these amino acid interbridges, or cross-links. And the cross-links always occur between the NAM in one chain and the NAM in an adjacent chain. Penicillins typically will interfere with the formation of this amino acid interbridge right here, making the cell wall structure very fragile and loose. Now, there's some interesting things in this interbridge. There's some unique molecules. And one of the unique molecules are some D amino acids. So you can see right over here, L-alanine, D-glutamine, L-lysine, D-alanine, lysine, D-alanine, D-glutamine. What do the D smells refer to? Stereoisomers. And amino acids can occur in both the D and the L forms. For dextrorotatory and non-rotatory. Okay. For some reason, natural selection favored almost exclusively L isomers in building polypeptide chains. So all the polypeptide chains that make up the structural and functional proteins in your cells are made out of L isomers of amino acids. But for some reason, in these inner bridges right here, the D isomers are also found, and that's very unusual. There's also a couple of modified amino acids. One, again, that is unique to prokaryotes. It's not shown on this particular slide. Because the nature of these inner bridges, what amino acids they have and how they're put together, varies from species of bacteria to species of bacteria. But one of the modified amino acids is this compound called DAP, diaminopimelic acid, and that is also unique to prokaryotes. So you've got some interesting molecules going on here that you only find in the prokaryotic domains. You don't find those molecules in eukaryotic cells. Okay, let's take a look at gram stain for a second. Give you a little preview of what we're going to be doing. The gram stain is a type of stain called a differential stain. And differential stains split organisms into various groups depending upon the outcome of the stain. Obviously, the two outcomes of this stain are gram positive and gram negative. Differential stains involve more than one staining reagent. The very first stain you're going to do is a simple stain, where you just stain cells with one staining reagent. Purple stain crystal violet, or the red stain saffron. The two primary stains in the gram stain are a purple stain, crystal violet, and a red stain, saffron. Another important ingredient in the gram stain is iodine. Iodine serves as a mordant. Have you ever heard that term before? Mordant is kind of like a cement. Think of kind of like mortar, the stuff that holds bricks together. And then a very important part of the staining process involves the use of 95% ethanol. "95% ethanol serves as what we call a decolorizing agent. So what this does is it shows you what the step is, what the appearance of both gram-positive and gram-negative cells are at that step, and then shows you what's going on in the cell wall itself to give you this particular appearance. Okay? So the first step in gram staining is to prepare some cells, get them on a slide, and then treat them first with the primary stain, crystal violet. Crystal violet is a positively charged stain, and it binds to the negatively charged cells and turns the cells purple. And if you look at their bacterial cell wall structure, the crystal violet stain is embedded into the peptidoglycan layer. You even get a little bit in the LPS layer right there. The next step involves then adding iodine. And basically what iodine does is kind of precipitates the stain and kind of holds it in place. So the appearance of the cell after iodine treatment is the same as it was the previous step. Both cells look purple. And here we've got dye crystals trapped in the cell. Okay? So what we'll pick up next time is what happens when you decolorize, and then why do we end up with the red and pink cells when the process is done. Okay, so see you at 9 tomorrow.